What is the best solution? Ferrari or Tesla? It's difficult to choose. Ferrari is a fascinating Italian luxury car, rich in history with a seductive sound. It is simply a Ferrari. It uses gasoline as fuel, and it doesn't work without gasoline. Tesla uses electricity as fuel, and it is performing, technological, and silent. It doesn't work without electricity. In the course of our evolution, the organism has had to choose its fuel and adapt itself to various environmental fluctuations. And to do this, it became flexible. Who selects the fuel? The mitochondria. The mitochondria is our hybrid engine that is able to use both glucose and fat as fuel. The concept of metabolic flexibility was particularly linked to the capacity of mitochondria to select fuel in response to nutritional changes. But there is a problem. Humans switched their primal lifestyles towards a plethora of energy-dense foods, persistent psycho-emotional stressors, and lack of exercise in a record time. All these changes favor decline in metabolic flexibility and favors the development of several metabolic diseases. In other words, metabolic inflexibility. And this promotes unsuccessful aging. In my presentation, I explore the concept of metabolic flexibility based on newly emerging data, and I give you some suggestions to improve it. Metabolic flexibility is the ability of cells to switch from a state of predominant lipid oxidation during fasting conditions to the suppression of lipid oxidation and decreased glucose uptake, oxidation, and storage under insulin-stimulated conditions or after a meal. During the postprandial period of a carbohydrate-rich meal, pancreatic beta cells respond to the rise in nutrients by releasing insulin into the bloodstream, increasing the insulin-glucagon ratio. Under the influence of insulin, the liver is triggered to absorb glucose from the circulation and stop glycogenolysis and gluconeogenesis. Skeletal muscle assists in glucose clearance as insulin receptor binding of insulin results in translocation of glucose transporters, such as GLUT and particularly GLUT4, to the plasma membrane, allowing glucose to enter the cell. Adipose tissue responds to insulin by decreasing the rate of lipolysis and stimulating fatty acid synthesis from lipids and glucose. During fasting, the decrease in circulating dietary carbohydrates and lipids and the decline in insulin-glucagon ratio induces a switch toward fatty acid oxidation. Glucagon stimulates hepatic glycogenolysis and ketogenesis, and the decrease in insulin suppresses malonyl-CoA synthesis and lipogenesis with concomitant activation of fatty acid oxidation. Because the adipose tissue is the predominant source of free fatty acids, the capacity of adipose tissue to store fatty acids during caloric availability and release fatty acids during caloric restriction is an important determinant of metabolic flexibility. What happens with caloric excess? With caloric excess, the mitochondria are overwhelmed by an excess in substrates derived from fatty acids, glucose, and amino acids. This surplus leads to mitochondria metabolic indecision, resulting in incomplete substrate utilization for energy production and consequent storage of substrates in ectopic depot. In conditions of substrate overload, the mitochondria must make a choice between acetyl-CoA delivered by either pyruvate dehydrogenases or D-beta oxidation. This results in elevated levels of acetyl-CoA and other acetyl-CoA at crucial sites where the catabolism of fatty acids, glucose, and amino acids converge, leading to a state in which upstream substrate catabolic flux slows down considerably, a phenomenon coined as mitochondrial metabolic gridlock. Skeletal muscle, just a flash. We know that during low-intensity exercise, oxidative muscle fibers predominantly rely fatty acid oxidation. During more intense exercise, the rising ATP utilization rate induces a switch from fatty acid oxidation to glucose metabolism. During acute exercise, an increased AMP-ATP ratio sensed by AMPK increasing the activity of the PGC1 alpha, that is a major regulator of exercise-induced adaptations. Metabolic flexibility is negatively correlated with aging. Several age-related changes, such as mitochondrial dysfunction, muscle deterioration, cellular senescence, hyperinsulinemia, and inflammation, affect metabolic flexibility simultaneously. Systemic inflammation and metabolic inflexibility can cause a vicious circle, because metabolic inflexibility can also trigger systemic inflammation, meta-inflammation, metabolically trigger inflammation, then inflammaging. To maintain energy homeostasis, different tissues secrete factors such as adipokines, myokines, hepatokines, and patokines. We know that adipokines such as leptin and adiponectin provide signals in response to changes in energy storage. The adiponectin-leptin ratio is used as a biomarker of adipose tissue dysfunction. Myokines play important roles in mediating the positive effects of exercise on metabolism. Myokine act as a communication signal for skeletal muscles to regulate the whole body metabolism during exercise. For example, exercise decreases the expression of myostatin in humans, and obesity is associated with increased myostatin expression. Hepatokines such as FGF21 and phytoin can also regulate metabolic flexibility. For example, phytoin has a major role in the regulation of insulin sensitivity, as phytoin deficient mice showed improved insulin sensitivity. Batokines produced by the brown adipose tissue are involved in the regulation of body temperature and energy expenditure via thermogenesis. Now, we explore how metabolic inflexibility can be reversed by lifestyle and pharmacological intervention. Your mitochondria are what you eat and depends on your diet. Historically, this area of study has been divided into opposite viewpoints that argue for or against the idea that it is healthier to burn fat than glucose. Recently, these two camps have found common ground in the concept of metabolic flexibility, which posits that cells function optimally when they retain their capacity to switch freely between oxidative substrates in response of nutritional and physiological cues. Importantly, a certain dietary intervention will have different effects on different individuals. In other words, different individuals may follow different trajectories toward improved health, positive aging, and longevity. These trajectories may depend, for example, on individual epigenetic regulation, human constitution type, lifestyle, stress, and on the national function level. Moving beyond the standard recommendation to eat less and move more, we have personalized nutritional approach, including intermittent fasting and time-restricted feeding, on which there is a plethora of newly emerging data. Both habitual aerobic and anaerobic activities promote considerably health benefits, such as increased mitochondrial content and metabolic flexibility. Don't forget that cold exposure as well as circadian clock can favorably influence metabolic flexibility. The pharmaceutical approaches to improve metabolic flexibility have been studied in great detail. Several evidence suggests that boosting the capacities of the systems that mitigate mitochondrial stress, such as antioxidant defense, glutathione, carnitine, NAD, AMPK, and sirtuin, Activators, for example, provide a useful strategy to stimulate metabolic flexibility and control energy homeostasis. Mitochondria is what we eat. We must absolutely avoid mitochondrial indecision as well as mitochondrial metabolic gridlock, preserving the ability of mitochondria to select fuel in response of nutritional changes. We have to be flexible. So don't choose between Ferrari or Tesla. We have many solutions to move and feel emotions. Thank you for your attention.